Today, an FDA approval in multiple myeloma, a priority review designation in diffuse large B-cell lymphoma, an application accepted for a biosimilar, encouraging findings in multiple myeloma and Hodgkin lymphoma, and disappointing results in acute myeloid leukemia. Welcome to Enclave News Network. I'm Gina Columbus. The FDA has approved isatuximab IRFC, known by the trade name Sarclisa, for use in combination with pomalidomide and dexamethasone for the treatment of adult patients with multiple myeloma who have received at least 2 prior therapies, including lenalidomide and a proteasome inhibitor. The approval is based on results from the Phase 3 ICARIA-MM trial, in which the addition of isatuximab to pomalidomide and low-dose dexamethasone led to a greater than 40% reduction in the risk of disease progression or death versus pomalidomide and dexamethasone alone in patients with relapsed refractory disease. Moreover, the objective response rates were 60.4% and 35.3% respectively. At a median follow-up of 11.6 months, the median progression-free survival per independent review was 11.53 months with the isatuximab regimen, compared with 6.47 months with PD alone. While overall survival data were immature at the time of the analysis, there was a trend toward improved survival for the isatuximab arm. The PFS benefit held up across multiple patient subgroups. The median OS was not reached in either arm. The 1-year OS rate was 72% with the isatuximab triplet, compared with 63% with pomalidomide and dexamethasone alone. In diffuse large B-cell lymphoma, the FDA has granted a priority review designation to a biologics license application for the combination of tafasitamab and lenalidomide for the treatment of patients with relapsed refractory disease. The application is based partly on findings from a primary analysis of the L-MIND trial, which showed that the combination elicited an objective response rate of 54%, including a 32% complete response rate as assessed by an independent review committee in this patient population. The BLA is also based on results from ReMind, a retrospective observational matched control cohort evaluating real-world efficacy outcomes of relapsed refractory patients with DLBCL who received single-agent lenalidomide and compared them with the L-MIND data. The FDA will make a decision on the application by August 30th, 2020. The FDA has accepted a biologics license application for a proposed biosimilar for bevacizumab. Mylan, the company that develops the biosimilar with Biocon, stated that the application is under review by the FDA with an action date of December 27, 2020. The company also announced that it had submitted a European application for the bevacizumab biosimilar that is currently in the validation stage with authorities. Mylan and Biocon co-developed the bevacizumab biosimilar MYL14020, known by the trade name Abevmy, which was approved by the Drug Controller General of India in November 2017 for all indications of reference bevacizumab, including as a treatment for patients with metastatic colorectal cancer, non-small cell lung cancer, glioblastoma, ovarian cancer, cervical cancer, and renal cell carcinoma as part of specific regimens for Indian patients. Results of a single-center, randomized, double-blind, 3-arm parallel group study evaluating MYL14020 also demonstrated biosimilarity between the biosimilar and both EU Union-sourced and US-sourced bevacizumab. In multiple myeloma, top-line findings from the Phase 3 Boston trial showed that selinexor in combination with bortezomib and low-dose dexamethasone led to a statistically significant increase in progression-free survival versus bortezomib and dexamethasone alone in patients who have received 1 to 3 prior lines of therapy. Results showed that the median progression-free survival in the selinexor arm was 13.93 months, compared with 9.46 months with bortezomib and dexamethasone alone, leading to a 30% reduction in the risk of disease progression or death. Additionally, no new safety signals were reported with selinexor, and there was no imbalance in deaths between the 2 arms. Full findings will be presented at an upcoming medical meeting. In July 2019, the FDA approved selinexor for use in combination with dexamethasone for the treatment of adult patients with relapsed refractory myeloma who have received at least 4 prior therapies and whose disease is refractory to at least 2 proteasome inhibitors, 2 or more immunomodulatory drugs, and a CD38-targeted monoclonal antibody. Pembrolizumab demonstrated a statistically significant and clinically meaningful improvement in progression-free survival compared with brentuximab vedotin in adult patients with relapsed refractory classical Hodgkin lymphoma, meeting one of the dual primary endpoints of the Phase 3 KEYNOTE-204 trial. The top-line findings were part of the interim analysis that was conducted by an independent data monitoring committee. Per a pre-specified analysis plan, overall survival, which is the study's other dual primary endpoint, was not formally tested at this analysis. The study will continue to evaluate for OS. Moreover, the safety profile with pembrolizumab was found to be consistent with prior studies of the PD-1 inhibitor, and no new safety signals were identified. Full findings will be presented at an upcoming medical meeting and will also be submitted to regulatory authorities. In acute myeloid leukemia, results from the phase 3 Vial-C trial showed that the combination of venetoclax and low-dose cytarabine did not lead to a statistically significant improvement in overall survival versus low-dose cytarabine alone in newly diagnosed patients who are ineligible for intensive chemotherapy. Data showed that the combination did lead to a 25% reduction in the risk of death, with a median OS of 7.2 months compared with 4.1 months for LDAC alone. However, this was not found to be statistically significant. Moreover, a post-hoc analysis after an additional 6 months of follow-up showed a median OS of 8.4 months with the combination versus 4.1 months in the control arm. In November 2018, the FDA granted an accelerated approval to venetoclax for use in combination with azacitidine or decitabine or LDAC for the treatment of adult patients with newly diagnosed AML who are at least 75 years old or who have comorbidities that preclude use of intensive induction chemotherapy. This week, we sat down with Dr. Sergio Giral of Memorial Sloan Kettering Cancer Center to discuss clinical trials of narsoplimab in the management of transplant-associated thrombotic angiopathy.
So Dr. Lawrence et al., who's been an expert in the field, has been conducting a prospective study of patients undergoing an allogeneic transplant and looking for those who develop criteria for transplant-associated TMA. He's been looking for different biomarkers, and in the abstract that he presented in the American Society of Hematology meeting, he specifically reported on the levels of MASP2. MASP2 is the enzyme that actually does complement activation through the lectin pathway. What he showed is that all patients undergoing allogeneic transplant had high MASP2 levels. But interesting, the patients who developed transplant-associated TMA actually had higher MASP2 levels than the controls. He also showed that patients with persistent transplant-associated TMA that did not resolve despite withdrawal of the offending agent or treatment of the infection had continued elevated levels of MASP2. What this suggests is that MASP2 may be involved in the pathogenesis of transplant-associated TMA and therefore is an appropriate target for therapeutic intervention. Rambaldi et al. Presented in the European Society of Hematology a very interesting preliminary results of a phase 2 trial of narsoplimab for patients who developed transplant-associated TMA. It was a dose escalation study, so it was a dose-finding study. A total of 19 patients were treated, 18 were adults that had transplant-associated TMA. What was important is that in this study, in all patients who received narsoplimab, there was at least some objective evidence of improvement in the evidence of transplant-associated TMA. That means that creatinine improved, LDH decreased, hemolysis and platelet counts improved. More importantly, the primary endpoint, which was 100-day survival, was significantly better for patients receiving narsoplimab versus historical controls. That's 53% versus 10%. This suggests that narsoplimab might be a very effective agent for the treatment of transplant-associated TMA or for the prevention of endothelial damage, and those are subjects of current and future studies. The inclusion criteria was to be within 30 days of transplant, have criteria for transplant-associated TMA based on the standard criteria, decreased platelet counts of less than 150, doubling of creatinine, increased LDH, and presence of schistocytosis in a peripheral blood smear. The Rambaldi trial was very important because the 100-day survival, which was the primary endpoint, was 53% for the patients treated versus 10% for a group of historical controls. We need to remember that currently there is no drug that has been approved for the treatment of transplant-associated TMA in the United States. The group in Cincinnati has pioneering and very effective data showing that eculizumab, which is an inhibitor of complement activation that does not go through the lectin pathway, has been effective in the treatment of transplant-associated TMA in children primarily. The results in adults have not been as promising, but again, it suggests that transplant-associated TMA in children might have a different pathway than transplant-associated TMA in adults. The narsoplimab data that was provided by Rambaldi et al. Suggests that maybe the lectin pathway may also be involved in this transplant-associated TMA. I think as we move forward, we are going to see that one, there will be more awareness of transplant-associated TMA. Two, we will be looking more at markers of complement activation, such as CH50, C5b9. And three, that many of us will be participating in collaborative trials of either narsoplimab or eculizumab or other complement inhibitors to be able to prevent this devastating complication of allogeneic transplant. Actually, most of the experience with transplant-associated TMA and a lot of the what we've learned from transplant-associated TMA has emerged from the group in Cincinnati, Stella Davis, Sonata Giondelli, and Chris Dunley. And what they've done is they've shown, when they look for transplant-associated TMA, they actually see it in 30% of the patients, of which a third can have severe transplant-associated TMA. They also have very clear algorithms for the use of complement inhibition, and they actually monitor complement activation throughout the transplant course. And this has resulted in their reporting the beneficial effects on eculizumab as treatment for transplant-associated TMA. Similar results have not been observed in adults, although there are cases that have responded to eculizumab. Is transplant-associated TMA more common in children and adults? The reality is we don't know. Prospective trials are now currently being performed to get a better handle. One of the problems is that schistocytosis, which is a hallmark of thrombotic microangiopathy, is not a very trustworthy test. I mean, you can get the same slide being read by different people and different number of schistocytes.
That's all for today. Thank you for watching Enclave News Network. I'm Gina Columbus.